thank you. And Sangoro is here with us. Very exciting. So we do have, I think, most people, which is very, very exciting. Sangoro, could you try just sharing your screen just to make sure that it's all working? Thank you so much.
Yeah. Give me a minute. You can continue.
Okay. If people are happy with it, I'm going to just start showing some of the surveys and such whilst Sangoro sets up. So let me share my screen as we kick things off. So again, as Sangoro will set things up, I'm just going to kick things off very quickly. So welcome everyone to workshop 2. We're very, very excited. Sangoro will be leading this session about introduction to malaria. But today we're just going to start with how amazing these surveys are that you guys have been spending a little bit of time filling it out, and how all this information is really, really helping us to adjust our materials and just get feedback. So again, thank you so much for everyone for filling these out. And I thought I would just show that these were, for example, the pre-launch survey results. And as you can see, these were kind of like what is everyone's experience with different software and such. And we really, really appreciate this. So we can see that some people have some experience with like R and GitHub and such, but quite a few people have not used these before, which is fantastic. And that's why we are kind of aiming to have lots of introductions, and then we do advanced stuff as well. And as you can see, it's all anonymous, but it's providing us with really cool data, again, informing us how to put together our presentations and such. And I thought I would show that we have lots of amazing, leaders in our group. Well, I know it says somewhat confident, but I would very much like to encourage you guys again that if you would like networking sessions, if you'd like to have more one-on-one time with facilitators or something, just let us know. And again, this is your program, so feel free to take leadership of it and everything. And we have really good communicators within us as well. So it's going to be very exciting when it comes to the communication and writing section as well. And I thought I would show that this was before the first workshop about what is version control. Then when people described it, we could see that people were familiar with the descriptions as well, and then how the percentages have changed after the workshop itself. So all of this is very, very, very useful. Again, pre-workshop 1 survey, how confident are you? Lots of people usually say somewhat confident, and look at this. Lots of people have become very confident, which is fantastic to see. So hopefully, again, we have been able to deliver impactful workshop and such. And you can see here as well about using functions and how these things change over time, which is, again, just really, really useful information. And before this workshop, thank you so much for filling out the surveys. I'm just going to put it in the chat again, the link to our Google Drive, where we are taking collaborative notes as well. So I'm putting extra information there. Feel free to add anything if you want to share. And you can see that with how confident are you describing the biology of malaria and different species. And most people said not confident. That's fantastic. Genuinely, we really, really appreciate all this honest insights, and really hope that after this workshop, the blue stuff, the nose will shrink, and most people will say that, oh, now we know what all of this is about. So again, I thought I would just show, and if you guys would like to see some results from surveys from like workshop to workshop, I'm really happy to share. Again, I'm a really big survey nerd and everything. And as we have mentioned at the beginning of the this session, and thank you for everyone for joining us. Very happy to schedule a networking session. I sent out a calendar invite for Friday. Please feel free to let me know today, or just text or write in the chat or something, like when it would work. It was fantastic to see that everyone was like, yes, everyone is interested in like just a networking session where people have time and we're not in a rush. So thank you so much. So 3 people have sent me screenshots of the different tasks and everything. And I thought I would just mention that thank you so much for sending this. I think I was able to spot a very few things, and I hope that everyone has been able to go through the different Excel tasks. As I mentioned it in the WhatsApp as well, I have recorded on like screenshots and description of the task, and I put it on the website itself, embedded these videos. So hopefully those were useful information. And again, that idea came from a survey. So thank you so much for the suggestion, whoever put it forward. And whenever we have a workshop and there's some material or resources that would be really handy, just give me a shout and I will try my best to make that happen. So just to mention, I think the NA ignore, there are different ways of going about it. I think one person literally just got rid of the NAs and left the cells empty. We don't want to do that because we really want to we want to keep the NAs in their cells, because it's going to tell us information that the information wasn't available or wasn't recorded in that month at that health facility. If you delete it, it could actually become like an error that we introduced or someone else introduced, and we're not quite sure where the empty cell has come from. So there is a function or formula that if you define if NA, and then you select the cells and you put the 2 parentheses, it's going to treat those cells as empty. So it's going to ignore when using the sum function. So I thought I would mention this. I hope this is useful. If anyone had any issues, just please let us know. With the VLOOKUP, I think people have been sending me screenshots not using the $sign at the first cell. And I thought, you know, it would be great to hear people's experiences, because when I tried that, when we moved on to admin 2, the numbers were jumbled up because that little $sign at the beginning, it means that we keep on wanting to use that cell as a reference when we're matching up with the table array, so from the population tab or column and such. So I would just like everyone to double check that it has worked. If it has worked with the $sign, without the $sign, that's fantastic. I don't know, for me, it was really, really needed. And with the TextMe or the README text files, I think a few people mentioned that it wasn't quite clear what we were trying to achieve, and that was mostly because we didn't have time to go through it in time. That we want to, as we're making changes to the CSV file, we want to document what steps we have taken, and then you save the text file or the CSV file, and you put in the CSV file name changes into the text file. On the website, it's also kind of described that like we would have routine data.csv. Then we would need to save it as routine data like underscore clean.csv. And in the text file, then we would just document like what has happened to it and how we have renamed the CSV file. Because when we have, for example, lots of different outputs on different dates and everything, we need to still identify what is the difference between these dates or different files and such. So again, one of the videos on the website that I've recorded, I went into a little bit more detail. I hope that helps, but if there's any more clarification why we need these README text files and such for explaining what's happening to, for example, a CSV file or something, please let us know, and we can keep chatting about this. So I hope that this was a good kind of way of starting. And if Sangoro is ready, I will start handing over. But if anyone has any questions about the Excel task or anything, either maybe mention it now. Please feel free to unmute, put your hand up and ask the question, or at the end of the session as well. I'm really happy to stay here and have chats if anyone has any questions. Thank you so much. I'm going to stop sharing. Sangoro, are you ready to kick things off? Thank you, Kokoda, for the reaction. Has everyone been able to complete the task, the Excel task and everything? Or maybe in R as well, whoever might have had the experience? Please feel free to share your experiences, and Sangoro as well, let us know once you have sorted out your slides, and then we can kick things off.
Could you repeat what you asked?
Could you repeat it, Kokoda?
You mentioned something if someone has experience, but I didn't get it.
Oh, probably the $sign. That I think on your screenshot as well, you didn't use the $sign for referring for the index value, which is fantastic. I don't know why. For me, the $sign was really needed, but then for you, it worked without it. Thank you, Kokoda. Okay,
hello,
Sangoro.
Jennifer. Can you hear me?
Yes, hi, Oscar. How are you doing? We can hear
I'm doing
you.
great. I'm doing fine.
Good.
Yes, I want to add on the using of VLOOKUP when you put $sign. Yes, it works perfect on my side.
Ah, cool. Fantastic. Did you find the task and everything okay? Otherwise?
Yeah, it was okay. Yeah, I found it okay. And I sent some of the screenshots, and I get some of the issues that you have been able to spot on my works, and I'm still working on them. Perhaps after finishing this workshop today, I'll be able to send the correction. Yeah.
Thank you. Yeah, I spotted a few things. Again, everyone, please send me screenshots and I can spot a few things, maybe in your Excel sheets or anything, if you have come across anything. And thank you so much, Oscar, again. Okay, so I think the one and only Sangoro is ready. So I'm going to hand over and yeah, kick it off.
All right. Good morning, everyone. So today we're going to go through intro to basic epidemiological concepts and an intro to malaria epi. So we're going to start with basic. So this workshop has two parts. We have the general epidemiology, and then we have intro to malaria epidemiology. So for the first part, introduction to basic epidemiology, we'll need to, by the end of this session, I hope you'll be able to define and describe basic epidemiological concepts. You'll be able to describe different types of epidemiological studies, factors that influence disease epidemiology, measures of disease frequency. You can be able to tell different study designs, measures of association, and interpreting the study results and findings. So What is epidemiology? Epidemiology is a study, or it's the study of distribution and determinants of disease. So in this workshop, you're going to hear certain words that we're going to repeat over and over again. That is what forms the basis of epidemiology. So the definition of epidemiology is that it's the study of distribution and determinants of disease, and in a specified population, and the application of this study for the control of diseases. So epidemiologists usually like to think themselves as detectives. So our work is to, like I've said, study the distribution of disease. So and then study the distribution of disease is like investigating how, I would say where, when, and who has the disease. And then determinants is to, as an investigator, it would be the culprits, or the agents that are culpable for causing the disease. And then, of course, arresting it would be to stop the disease. So to conduct epidemiological, in epidemiology, the investigators, or if you will, the detectives or epidemiologists as such, usually have tools in the toolkit which help them to carry out the study or to investigate disease. So, sorry, I started off quite wrongly, but this is going to be an interactive process. So I will need you guys to really interact so that we go faster. We go as quickly and as smoothly as planned. So can you guys think of any, and I have your names here, so I hope you'll participate without me calling your name, but can you guys think of toolkits that epidemiologists would have in their toolkit that would help them to investigate disease? Anyone? I'll start with Phoebe. Can you think? Okay, cool. Thank you.
So one of the tools, in my opinion, I would say that start the distribution of the particular disease. First, you need to have the information regarding the population you are studying. So which means you have
So the information, yes, about the population. All right, good. What other thing what other tool would an epidemiologist need? Anyone?
Can I add up?
Yes, please, Oscar.
So I might not have a background of epidemiology, but I can try. Maybe reference materials such as epidemiology textbooks can be used as a tool also.
Okay. Cool. Anyone? Anything else? Yes, Harriet.
I'm thinking measures and numbers.
Measures and numbers. Okay. Anyone else?
Historical data, either of the disease that they want to investigate more about, or even just like other diseases.
All right, cool. So yeah, you're all correct. So like Kokoda said, you need to have the information, and this information, like Neema and Harriet and Oscar have said, you need to have the information, and this information you can either use historical data, or you can collect the data using things like, so a tool would be something like a questionnaire, yeah, where you take to the community and then collect the information about a particular disease or event, yeah. And then, so that would be to assess the distribution. So if you heard that in Mkopeka, in Nyimba in Zambia, there's an outbreak of leishmaniasis, yeah, you go to Mkopeka and ask and use a questionnaire: who has the disease, when did they get the disease, what are the activities and residents. So that is one of the tools. The other one would be for a determinant, a basic one would be like a microscope, yeah? So you collect sample from people from the population and then look at them under microscope to be able to determine if they have an organism or an agent causing disease. Yeah, so I'll try and move a bit faster. So yeah, so basically epidemiologists would like to know what is happening, where, why, how, when, and what they can do about the disease. And to do all this, they first need to collect the information. Thank you. So using our first exercise. So the guys who haven't talked, I need you to talk. So using the key terms that I've outlined in the definition, can you match the following key terms with the activities that have been given? So the first activity, so we have the key terms are distribution, determinants, and application. So the first activity would be you're asked to map on the map the houses of children with kwashiorkor. Anyone? Let's see, Mwinde. What activity is that, and what key term does it match to?
I think he's not on the call yet, by the way.
Ah.
So sorry.
Phoebe?
I think I can match with determinants.
So if you're asked to mark on the map the houses of children with kwashiorkor. Say that, Kokoda?
Can I say?
I didn't hear you properly. Repeat, please.
I would say distribution of
Yes, distribution.
kwashiorkor.
All right, cool. Yes. The second one is recommend close contacts of a man with COVID-19 to receive AstraZeneca vaccine. What are you doing there in terms of epidemiology?
I think that's application.
Application. Thank you very much. So you have the information that if you understand the epidemiology of COVID-19, and if you separate them from a close contact person, then, or if you give a close contact person a vaccine, then you prevent them from getting COVID vaccine. That is application of the information you have about COVID. Compare sources of water of people reported to have diarrhea. Who? Is Jacqueline on the call? Hello, is Jacqueline on the call?
She's not, sadly, no.
Okay, Harriet, you'll you'll be put on the spot today. I know your name, so
Yeah,
that
that would be determinants.
That would be
Determinants.
all right. Cool, correct. Number of men with obesity between vegetarians and non-vegetarians. Name.
I think that would also be determinants.
Oh, very correct. Thank you. So, what are the aims of? Oh, we're gonna move. The aims of epidemiology, as you've seen, it's to describe the distribution and magnitude of disease. So, like Okoda said, distribution of kwashiorkor in that village. Identify factors that lead to the transmission of disease, determinants of disease. So, obesity, like we've seen, is either if you're not. In this context, would be say if you're not a vegetarian or if you're a vegetarian, that's a determinant of obesity. And then provide data essential for planning and implementation. That would be application for the disease control, which is AstraZeneca vaccine. So we're going to go through the history of epidemiology a bit quickly, I hope. So for ages, people have tried, or scientists, philosophers have tried to describe how disease is spread, the historical description of disease, of spread of disease or transmission of disease. So there are several theories that exist for the transmission of disease. I think one of the most common and most historical, or the earliest one, was religious beliefs, where people believed that if you are a bad person, for example. The gods would send you disease to punish you. So an example of this that comes to my head would be the times of, I would say, Moses, when the plagues were sent to Egypt to make Pharaoh to let the people of Israel go. I think that is one of the most common instances I can think of religious belief as a cause of disease. And then we came to the period of the miasma theory. Hello? Okay. Yeah, so we came to the period of the miasma theory. This is in the early 17th and 18th century, where philosophers and scientists believe that you got disease from inhaling putrid air. Putrid air is air that, smelly air or contaminated air or air that smells bad. So if you inhaled maybe air that belonged to air that came from a rotting carcass, then you'd catch disease. And then came the period of germ theory, where philosophers believed in that diseases were caused by microorganisms that infected an individual, a person. And this, I think, has been historically and contemporary what has been proven to be the cause of disease. So in the 18th century, in London, in a place called Soho in Broad Street, London, there was a cholera outbreak in 1954. And a lot of people are affected. And in this time, there's a physician and an anesthesiologist called John Snow. So John Snow was a proponent of the germ theory, where he believed that diseases were results of human beings being infected with microorganisms, which caused them to be sick. But of course, as with everything at that time, everything was a conspiracy. So people did not believe what Johnson was saying. So he set out to try and prove his concept. So John Snow realized that in a river that was next to Soho, they had let sewage run into the river, so in essentially contaminating the river that was next to Soho. So John Snow hypothesized, or no, let me not choose hypothesized right now. So John Snow theorized that people got cholera from the water in the river, which somehow got into which somehow got into the sources of water that people were using for drinking and cooking in Soho. So to set out to prove his theory, he mapped. You can see he mapped cases of cholera in London. So basically, majorly it was in Broad Street, London. He mapped out these cases, yeah? And also, because his theory was that people got it from infected water, he mapped out the sources of water. So you see here, pump A, pump B, pump C. So people used to get water from the pump. So his theory was that somehow the water from the river contaminated this water pumps, and this is what caused the people to be sick. So he mapped out both the pumps, the sources of water, and the individuals who had the cases who had cholera. So then he went out and interviewed them. And the people that he interviewed around a certain pump, this is pump A, he found that they mostly got their water from that particular pump. And the people that got water from other pumps, let's say pump C here, pump C, and this pump X, he found that these people did not have essentially cases of cholera. So he went further and asked, so where do the sources of this water come from? And he found that like for this pump X, he found that there was a well, there was a brewery around there, and that brewery had dug a well. So the well was not in contact with the river, with the water from the river. So it was isolated, and people around this pump did not catch cholera. So in that way, John Snow then hypothesized that most the people who got cholera are people who mostly got water from pump A. So he went to the municipal council and told them, if you remove pump A, then I believe that the cholera cases would go down. And of course, like I said, in that time, he was labeled a conspiracy theorist before the pump was removed. After it was removed, then the cholera cases died down. And so up to today, we call John Snow the father of epidemiology, of modern epidemiology, because he was able to link the source of water, which was as a cause of disease for the cholera outbreak. And if you go to London right now, you can see the John Snow pub if you want to go have a drink. And the pump is still there, I think. Yeah, so we move on. I hope that is clear. So our second exercise would be from John Snow's cholera theory investigation. What factors do you think were necessary for the spread of disease, for the spread of cholera? Mwinde, I'm going to call you out. I hope you understood the story.
Yes, the Jon Snow story.
Yes. So what factors do you think were necessary for there to be spread of cholera?
Sorry, just to come again with the question. What factors?
What factors do you think were necessary for the spread of cholera in John Snow's
Or
story?
the factors that exacerbated the spread of cholera?
Yes.
Okay. Number one, one I would say contaminated water.
Okay. Thank
Water
you.
itself.
Okay.
I do presume that in those days, the issues of personal hygiene were not as important as it was, though it could be a factor if we look at the context of the modern epidemiology.
Okay,
And
thank
then
you.
we can also move in to look at the epidemiological triad. And this is the hosts, the pathogen, and the environment.
Okay, thank you. Anyone else? Harriet, Kokoda, Phoebe, Nema, any other factors that you think were necessary? Guys?
Also, contamination.
Okay.
Yes, contamination of water from the river.
Okay, cool. So yeah, thank you everyone. So if we move, I think Winde preempted me, but then in epidemiology, we have something called the epidemiological triad, or the model of disease transmission. Yeah, this epidemiological triad brings together factors that need to come to interact for disease to occur. So one of the factors is the infectious agent. Yeah. So the infectious agent in Snow's story was what? Let's see, Oscar. Hello.
Hello, can you come up again?
The infectious agent in Jon Snow's story. What was the infectious agent?
Infection agent.
Say that.
Is that for everyone?
I asked Oscar. Hello. Yeah, so Kokoda, what was the infectious agent? Can you hear me?
Yeah, I can hear you. I'm trying to think about it. Did you mention something like microorganisms?
No, it doesn't have to be, you don't have to name the microorganism. I think in this context, we can say the infectious agent, like everyone has said, was found in the contaminated water. And then
Yeah, that was the main agent.
yes. And then the host, what was the host in this case in Jon Snow's story?
The host.
Is it pump A?
Pump A?
Yeah.
Someone else?
I think it's the human beings, the people.
The human beings. Okay, yeah, correct. And then the environment.
The
It was the
environment
pump.
was water.
Yeah,
They
of the
contaminate
pump of
the
the
water.
water. All right. So, like I said before, we'll come back to the vector. Like I said before, for disease to occur, there has to be an infectious agent. Yeah, an infectious agent is an agent that infects a person and causes that person to have a disease. So it can either be biological organisms, and later we'll see the expansion of infectious agent has occurred because of time and definitions have changed. But an infectious agent is something, it's an organism that causes that disease to occur in a person or an organism. So in this case, the infectious agent was found in the sewage or the water from pump A. It did have the infectious agent. And an infectious agent has certain factors that determine whether it will cause disease or not. Yeah. So one of the factors that an infectious agent has is the infectivity. So in the infectivity of an infectious agent is the likelihood that an agent infects a host, given that the host is exposed to the agent. So for example, you know that there are some infectious agents or there are some microorganisms that if you come into contact with, then you get infected immediately. But then there are cases where you can come into contact with the infectious agent, but you do not get infected. Case in point, let's say if a child has measles, then it is very likely that he will transmit it to another child that he comes into contact with. This way we say measles is very infective. But if a child or if a person has leprosy, then you need a long contact time and more, as in a lot of contact with the person before the agent is transmitted from the infected person to the non-infected person. So that is the infectivity of an infectious agent. And that determines whether the disease will be transmitted or not. The other thing is the pathogenicity. So you might be infected with an infectious agent. Yeah, I might be infected with an infectious agent. But then, does it cause disease or not? So I can illustrate this in terms of, let's say, the normal flu. Yeah. If let's say the bacteria that causes bubonic plague, yeah, or plague, if you come into contact with the Yersinia pestis, I think, then you are very likely that you will get the plague, yeah. But then there are some agents that you're infected with, or they're intrinsic in you, but they do not cause disease. Case in point, let's say E. coli. E. coli is normal flora in the human being. Yeah. So in exceptional cases or in opportunistic cases where, let's say, the E. coli gets into the urinary tract, then maybe you can get a urinary tract infection. But normally it's in the body, but it doesn't cause disease. I hope that is clear. And then the other one is virulence. So the likelihood or how severe is the disease if you're infected with an infectious agent. So for example, you've heard the word virulent. There's some infectious agents, viruses, bacteria, or parasites that you've had are very virulent. Yeah. Virulence means that it causes a lot of damage when it infects you. Case in point, let's say you've heard about Ebola. If you get infected by Ebola, hemorrhagic fever, and then you're dead after a couple of hours, yeah? But how many people get normal flu and they survive? So normal flu is not very violent. It's not very damaging to the host, but Ebola is. I hope those explanations are clear. So these factors determine whether disease is transmitted, whether it's infective or how infective it is, how pathogenic it is, does it cause disease or not, how virulent it is. Yeah, how severe of the disease does it cause? This is just a slide of some of the infectious agents. I think we'll move ahead because of time. And then in the epidemiologic triad, we also need a host. Yeah, I've said we need an infectious agent. But then if you just have the infectious agent and no host, then the disease does not transmit. So you need a host, you need an infectious agent and a host for the disease to be transmitted. So a host is an organism or a person that is infected with the disease. Yeah. So with the infectious agent. So the infectious agent comes into you and then it causes disease. That is what a host is. Yeah. So just like the infectious agent, there are certain factors that a host must have that are intrinsic to the host that determine whether they have disease or not. So can anyone think of any factors that make someone, a host, susceptible to a disease or an infectious agent? Anyone?
I think what comes to my mind mostly are what we term as comorbidities, like someone have other chronic infections, chronic conditions rather, that predisposes them to have low immunity, something like diabetes, hypertension. I think those mostly tend to put someone at higher risk of being susceptible to infectious agents.
All right, cool. Thank you very much. Anyone else? Any other factor you can think of? Let's see. Oscar, again. Any other factor you can think that?
Yeah, I think low immunity. Someone with low immunity. Yeah.
Anyone else? Kafwanka? Hello?
I'm the one who said earlier.
Sorry.
Yeah, but I can go again. I'm thinking also of, I think, okay, it's fine.
Neema, can you go?
Yeah, I don't think I have anything different because I'm thinking about like maybe also the genetic makeup and all of that, but I think it connects to the points that have been mentioned earlier.
All right, cool. Yeah, thank you. So some of the
Maybe
factors.
just to add on,
yeah,
there could be factors such as age, the age for someone.
Cool. Yes.
We can also say race in the case of malaria, geographical location vis-a-vis race, and sometimes we can also, in the case of non-communicable diseases, issues of lifestyle could account as well. Yeah.
Yeah. Thank you very much. You're all very right. I don't
Also,
even know
I can
why.
add.
Yes. Yes.
Also, some people who lack vaccination. Yeah, I think also that they can face that. Yeah.
All right. Cool. Yeah. So, like I said, you guys are already very conversant. Maybe we should stop the class now. Anyway, so we have things like immunity. So whether a person is immunocompromised or not, as has been said, age, gender. So you know the factors that the diseases which would attack one gender as opposed to the other. We have things like younger kids have lower immunity in the case of malaria, so they get more infected than not. And then their genetic makeup. So for example, like someone said, sickle cell would make you for a lack of better word, I would say immune. You're less predisposed to malaria if you have sickle cell as compared to people who do not have sickle cell because of the shape of the red blood cell. And then the factors like what is that enzyme that if you lack, then you are more predisposed to COVID vaccine. So this, I would like to mention just here that these host factors are intrinsic, and they're basically more related to infectious diseases. Later, we'll see other factors that, as someone has mentioned, are related to other diseases that are not infectious or communicable. Yes, the other factor that needs in the epidemiological triad that needs to be present for disease to occur is what? The environment. So if you have the host and you have the infectious agent, but then they don't come into contact, they do not interact. For example, then you will not have a disease transmission. Case in point, the people who got water in the Johnston story from another pump that was away from pump A. The host is, they are the host, yeah? They're present. And the infectious agent is present. It's just that they're not in an environment where they can interact. I hope that is clear. So the environment then determined must be suitable or must propagate interaction between the host and the agent for the disease to occur. And the environment includes a lot of things. Like we said, pump A would be an environment because it brought contaminated water in contact with, which has the infectious agent in contact with person. We have other things like, I would say pollution, temperature, which would propagate the development of an infectious agent or disease or vector. So an environment has to be suitable for the disease to occur. Okay, so what environmental factors can you think of? Anyone? Quickly. Environmental factors. I'll start with Winde. I think you can go, then everyone can follow after you. Because,
Water,
yeah, water.
air.
Water,
Oh, okay.
Air pollution.
air pollution, good. What else?
Climate conditions.
Yeah, environmental conditions that influence disease.
Oh yeah, I say climatic conditions, but I guess that's like
Climatic
all it.
conditions. Thank you. Who else?
And there is also go for overcrowding like prisons.
Yes, good.
How about
Yeah, so
temperature?
temperature, brilliant. So
Yeah.
the factors that influence, they can either be demographic factors, like someone had said, population. So if people are crowded, then this is an environment that influences quicker transmission of disease. Yeah. They can be environmental or climatic, temperature, humidity, rainfall, like we say in the case of malaria, breeding sites. So you're more exposed to disease. So these are some of the environmental factors. We will move. So in disease transmission, we said that in the epidemiological triad, we said that all these factors, environment, host, and infectious agent have to come together. But there are some cases where the host and the agent cannot come together. Yeah? So in this case, then what happens? Can anyone think of what happens in such a case where you cannot have direct interaction between the host and the infectious agent?
Then they'll need
Harriet. Yes. Yeah. Say that.
they'll need a vector.
They will need a vector. What is a vector? I didn't hear. Did you say something?
Yeah, I'm saying it's mostly an information.
Okay. So yes, they will need a vector. Thank you very much. And what is a vector? A vector is an organism that transmits, that I would say transmit. Is it too early to use the word transmit? That moves the infectious agent from the source to the host. Yeah. So it brings about interaction of the host and the infectious agent in areas in situations where this interaction does not occur naturally. Yeah. So, and it can do this in two ways. So there are two types of vectors, the mechanical vectors. So a mechanical vector would move an infectious agent from the source to the host, and the host takes up or ingests or is infected with the infectious agent as is. So case in point, Salmonella typhi. Who knows what Salmonella typhi is and how it's transmitted? Harriet. Daktari.
Can you hear me?
Yeah,
Can you hear me?
I can hear. Yeah, you're a bit breaking up. Yeah, go on. You're breaking up.
Let me type in the chat.
Okay. So Salmonella typhi is the bacteria that causes, the bacteria that causes typhoid, yeah? And it's carried by houseflies from the human stool that of a person who's infected with typhoid to, and it's brought in contact with the person who's not infected with typhoid, a susceptible host, if you will. So this is a mechanical transmission. It takes the bacteria from the contaminated stool, brings it to either food, water, or somehow, and gets it in contact with the host. And then when the host ingests this, then they get the typhoid. So that's an example of mechanical transmission. Biological transmission is a vector which transmits the infectious agent from the person, from the source to the susceptible host, but the infectious agent has to undergo transformation so that it's infected, so that the host gets infected. Yeah. So this is a biological transmission. So, for example, we would have what? Is it schistosoma, which is, or okay, a better example would be plague. So the rat flea ingests the bacteria from the rat. It undergoes transformation in the rat flea, and then when the flea bites an individual, then he gets the bubonic plague. This is biological transmission. The most common is malaria. This picks parasites from an infected host. It undergoes transformation or biological transformation in the mosquito, and then it's infected to the person. That is the vector. So there are some diseases that require vector for it to be transmitted. I had said earlier that the definition of disease initially was a person getting infected with an infectious agent. But as time has gone on, the definition of disease has changed to include whether, so right now it's any departure from normality of the human body from mental, physical, social, and psychological state. And so the definition of infectious agents also had to change to include these are the infectious agents. So we have nutrient agents. So, for example, if someone lacks nutrients like proteins, you have protein energy malnutrition, then you can get something like kwashiorkor. We have chemical agents. So if you inhale certain chemicals, let's say DDT, then you can get cancer. So DDT has been found to be carcinogenic. Then, because it's departure from normal state, things like mechanical agents, if you get into an accident and you broke your leg, then you are departed from the normal state, and therefore we can say an accident is a mechanical agent of disease. Then we have physical agents, things like temperature. And therefore, to include also other factors that are not communicable, we had host factors like demographic factors, yeah, as I had mentioned factors. And now this includes things like socioeconomic factors, socioeconomic characteristics. You know that sometimes because of their socioeconomic status, then people can be predisposed to certain diseases. So if you don't have money to buy protein, then you're predisposed, your children will be predisposed to I would say kwashiorkor, marasmus. Lifestyle factors. What kind of lifestyle do you lead? Your interaction. This also predisposes you to certain diseases. Like someone said, your eating habits would predispose you to certain conditions, maybe diabetes, coronary heart disease, or exercises and stuff like that. Yeah. What I forgot to put here was things like political factors. Yeah. So political factors are right now also considered as a factor of disease. So if, for example, you live in an area where there are no health facilities, just by that fact, then you would be more predisposed to someone who lives near health facilities because they can get treatment better than you. Yeah. And then the factors like psychosocial factors. So like I had mentioned earlier, religious beliefs, the communities or populations that believe we don't need to go to hospital, will get cured by some diet and stuff like that. So all these factors predispose you to disease. We're going to talk about the basic reproductive number. So the basic reproductive number is the number of cases that one infected individual can transmit to other individuals if those other individuals are susceptible. Are susceptible, that means that that they do not have, if they're exposed to the infectious agent or the disease, then they catch the disease. That's what susceptible means. I hope that's clear. So a basic reproductive number is, like I said, the number of cases that are transmitted from a primary case to a secondary case. And it depends on two major things. The contact rate, so the number of people that you come into contact with for the period that you're infectious. Yeah. So for example, a disease has a short, a very short infectious period, let's say 2 days. And then you come into contact with, let's say per day, you come into contact with 10 people. What would be the reproductive number of that? Kokoda, you're a mathematician. Hello? Hello? Yeah.
Sorry, what's the question?
So the question is, has everyone understood the basic reproductive number? So it's the number of cases from someone from a primary case that infects second. So if I'm infected with a disease, how many people can I infect? So how many people can I infect during an infectious, during the period that I am still infectious? I hope that is clear. Yes, Juniper.
Hi, Sangoro. So Nema actually put in a question in the chat. What is the difference between primary case and secondary case?
Yes, so primary case, let's say a primary case is, let's say I live here. I'm the first person, let's say in this house, I'm the first person in my house that is infected with malaria. That I'm the first person to get malaria in my house. I am a primary case. But so how many people can I transmit it to as one person? So the person that I transmit it to will be my secondary case. Is that clear, Nema?
Yes, yeah, that's very helpful. Thank you.
All right, cool. So if anyone has a question, because I can't see the chats, please just raise your hand. Okay, so yeah, so that is the reproductive number. So how many cases can I transmit if I'm infected? To how many people can I transmit to? So my question is, if the infectious period of a disease is 2 days, and every day I come into contact with 10 people, what is my basic reproductive number for that disease? Anyone?
I'm thinking it's 20.
It is?
I'm thinking it's 20.
Yes, it is 20 because every day I infect how many people?
10.
10. So I infect 10 people today and I infect 10 people tomorrow. I hope that is clear for everyone. Okay. So yeah.
Yeah, just
Yes.
So the definition here, I see that it was specified in an entirely susceptible population, and I'm assuming that's does make a difference. Is there a case of like partially susceptible population?
Yes. So for example, it has, so the entirely susceptible population here is just to, so like I had mentioned before, there are factors, host factors that would prevent you from getting a disease. So here we are trying to see if I get infected with the disease. So we will see this later. If I get infected with the disease, if a person does not have immunity to that disease, or if I stay in a place where people do not have immunity to that disease, how many people can I infect? We will see the reason why we use susceptible population in this case. But yes, it's an entirely susceptible population. And we will see why it differs depending on whether a person is susceptible or not in a minute. All right, cool. So we have cases where a reproductive number is 2. So for example, in the case of Ebola, so these different diseases have different reproductive numbers. So measles, let's say one person can infect 18 people. Yeah. So here you can see that the more people I infect, that means that's how, depending on the arrow determines how infectious a disease is. Yes. So if a disease has high reproductive number, that means it's more contagious than one that has low reproductive number. So why we talked about reproductive number here is because the major objective of epidemiology is to reduce the reproductive number of a disease. So for example, and this now, Nema, I think is where your susceptibility question comes in. So for example, if I have, let's say, if I live in Ifakara Health Institute, Ifakara, Kilombero Valley. So we know that malaria is endemic and most people have antibodies against malaria. So they have the parasite, but they're not sick. They've developed immunity to this disease. So if I live in Ifakara and a person who lives in the UK, where people do not have immunity to malaria, do you think that the reproductive number would be the same? Anyone? Does the question make sense? Yeah?
Yes, it will.
It will be the same.
It won't be the same.
It won't be the same. Why? Anyone?
Because you have modified
Yes. Yes, exactly. So the disease transmission will be modified, like Harriet said, because of how susceptible individuals are. So you see, in the UK, if I have malaria, then it would be easier for the next person that I infect to get malaria or to be symptomatic. But in Kilombero Valley, where malaria is endemic and people have immunity, they might have the parasite, and they don't have the disease, and therefore cannot transmit the disease to another person because the immunity is modified, and therefore the RO in the number of people that I can infect in the UK is different from the number of people that I can infect in the Kilombero Valley. And this depends on the host factors, yeah, how susceptible individuals are. It depends on the agent factors. So how effective are they? Are the infectious agent? So like we had seen before, the infectious agent, there are infectious agents that even though you come into contact with, they do not infect the next individual. So those factors will determine the RO. Yeah. And of course, the environment. Does the environment, do the environmental factors then propagate the interaction and the development of the disease? So the major objective of epidemiology is to reduce the reproductive number, either by increasing immunity, by changing the environment, or by keeping the human interaction contact, by breaking that contact so that disease is not spread. So that is why we were talking about reproductive number today. Yes, Osman.
Hi, Sangoro. Yeah, thank
Hey.
you, everyone. So I want to add something about the beta contact rate, because in your definition you say that it is contact rate by infected host, but I think it is not only infected host, because if the disease is transmitted host to host, or you can say it is infective host, but if it is a vectorial transmitted disease, the contact can be defined from the vector to the host.
Yes, thank you. So in this case, yes, you're very right. So there are diseases that are are determined by the contact between the host and the vector, and then by the host to the host. Thank you very much, Osman. Yeah, I will correct that. Yes. Any other question? Okay. So epidemiology, there are two main approaches to epidemiology, to the study of epidemiology. There's one called descriptive epidemiology and analytic epidemiology. What does descriptive epidemiology do? So descriptive epidemiology, I think, and we've seen this before, is basically tasked with describing a disease. So describing the distribution of a disease. So descriptive epidemiology is mainly who has the disease. So this is in terms of gender. Yeah, distribution by gender, distribution by age, by occupation. And then what are the symptoms of the disease? Yeah, it describes the disease. And then what are the geographic regions? What are the places that the disease is found? And the temporal distribution, the geographic distribution, yeah? So descriptive epidemiology is usually just to describe how, when, why, how and when a disease occurs, yeah? And the purpose for this is mainly for developing theories for why the disease occurs. So it developed theories for what we'll see for analytical epidemiology. So for example, like we said, mapping the number of houses that have kwashiorkor in a village, yeah? Analytic epidemiology is more invested in investigating the determinants of disease, yeah? So understanding the influence of, so we have schistosomiasis in Chirets in Zambia. Why? What are the factors that propagate the transmission of Chirets? So it explores the why and how behind the disease. Is it the climatic factors? Is it the human factors? Is it the age factors that are found here? Yeah. So it involves investigations and comparisons between exposed and unexposed, and to determine the reason for the disease. I hope that is clear. You want to take a break, or do we go on? Okay, 5-minute break. 3 minutes actually. We are back in. We're back in 11:15. Thank you.
Thank you, Sangoro. I think you really deserve a really big cup of water with everything. So thank you so much again. And thank you so much for all the engagement, everyone. That's absolutely fantastic. And the chat as well, just keep using it. It's all fantastic. And I think, like, again, this whole section was all about like building up what you need to think about, you know, when working with an infectious disease, and like all the different variables. Hopefully you're imagining things and everything. So I will then just hand it back over to Sangoro, who's keeping us all very much on time and everything, and then a few more minutes. Thank you so much.
Okay. Okay, so has it been 3 minutes?
No, you're doing amazingly. You're very much like so fantastic. Thank you so much again. I think there was a question in the chat by Harriet. So basic reproduction assumes that everyone is fully susceptible. And Osman has also replied, and you replied as well. So it's fantastic.
All right, cool. So I will assume everyone is back. Can you still see my screen?
We can see your screen. It says click to exit full screen at the moment in the middle.
Yay. What did I do?
Is it because maybe it's on another screen?
No.
Oh.
Okay.
I know that Kokoda is having network issues, but it's all good. Can you remove the bubble at the top? It says turn on reactions. So we can see that on the top. Yeah, that one. If you just click on X.
What is the X?
So what I can see is your screen, and at the top, where it says Safari and everything, it says a bubble that turn on reactions, and it says that Zoom has turned off reaction effects and such.
Okay. All right, cool.
If you can just click on that one.
Yeah, it says
oh, now we cannot see your screen.
All right. Okay, now?
Are you happy to share your screen again? Because it stops sharing.
Okay.
Fantastic.
All right, cool. Yeah. Okay, so we're gonna go on. So question. So you've collected data on so you want to know, okay, you've not collected, you want to know about distribution of diseases. How do you go about this? How did we say we go about getting this information? Okay, so I think we get this information from, in epidemiology, what we call studies. Studies are basically just data collection, ways of data collection, yeah? So you can do this through observations, yeah? So you can sit somewhere in Maputo, in a hospital in Maputo, and just note the number of people who come up with trachoma, yeah? So you see Sangoro came in at this time, he stays here, he has trachoma. The great Jailos has come in, Juniper, So it's basically just sitting at a place and observing and collecting or writing down your observations or collecting information about your observations. These are mainly done in, I would say, hospitals where you have case reports. So someone comes to a hospital and then you record their cases. And it usually includes a case-by-case person. So a person comes, you take the individual reports, and then later you collect it into something that we call a case series report. So this is in the case of a hospital. in some other cases, you can just go to a community and observe them. So like let's say the issue I raised about the example I gave about or let me give an example about having leishmaniasis in Kopeka in Zambia. So you just sit in Kopeka village and then you see what the behavior is. So you say, so lady from house number 1 went to the river at this time, collected water from this time. When she came back 2 weeks later, she started complaining about itchy eyes. A month later, she cannot see. So it's you take such observations, yeah. It's a method of data collection, and then later maybe then you can be able to see the sequence of events which led to the outcome. Those are observational studies. The other type of study is what we call a cross-sectional study. A cross-sectional study is basically data collecting data from a population at a given point in time. Yeah. So you go to, which is a better example. I go to Perth in Australia, yeah, and say, I want to see how many people in this city have, what's a good example of a disease in Perth, Juniper? Anyway, yeah, you're looking for a certain outcome of a disease. So you sample, let's say 100 people, and from all these 100 people, you take samples and then you try and test to see if you will find them with a certain outcome of a disease. So this is called a cross-sectional survey. So you go to a population, take a sample of people, and from this sample of people, test how many people have the outcome of interest that you want. And cross-sectional studies can be done. So apart from cross-sectional studies, there's the longitudinal studies where you can take samples at different time points. Let's say in a year you go to Perth and you collect the samples every quarter, so every 3 months or every 4 months, to see the outcome of interest that you are interested in. Yeah. So that is a longitudinal study, because you've done a cross-sectional study is at one time point, a longitudinal study is at different time points, looking for the outcome of interest that you're interested in. The other ways that we can collect diseases that through a method called case control studies. So case control studies is, like it says, case control. So you sample a number of people who have a certain case. Let's say in this case, a group of outcome of interest is cancer patients. Yeah. So you take a group of people who have cancer patients, and then you take a group of people who do not have cancer, and then you take their histories. Yeah. You go back and see in this group of people who had cancer, what has been there, what has been, let's say, what has been their exposure, what has been their behavior. Yeah. What have they been exposed to? Yeah. And then you compare it with the histories of people who do not have cancer. And therefore, in that way, in that comparison, you can say, this factor is found in these people who are cases as compared to these people who are controls. So you cannot, so drawing conclusion is a wrong term I use here. You can associate a certain factor with a certain outcome. I hope that makes sense. So then we have things that we call cohort studies. Yeah. So cohort studies is a way of collecting data where you have 2 cohorts. You take a sample of 2 different populations, and then you follow them and you see what they've been exposed to. So if one group has been exposed to a certain factor and another group has been exposed to a certain factor, and then after a period of time, you compare their outcomes. This is a case control. Difference between case control and case studies. Case controls are retrospective. Cohort studies are prospective. So you come from exposure to outcome. In case control, you come from cases and then you determine you assess their exposures. And then you can draw associations between the two groups and with a certain outcome or with a certain exposure. I hope that is clear. If there's a question, please put it in the chat. And then we have what we call the randomized controlled trials. So the randomized controlled trials are the holy grail of data collection in epidemiology. Randomized controlled trials are mainly used to assess interventions. And like this, like the term says, it's randomized. So it's a randomized control. So you randomly pick a group of people and then assign them to group A, and then you randomly pick another group of people and assign them to group B. So here's a question, and I hope you've been listening. Why is randomization important in this case? Let's see who was not. Phoebe, why is randomization important?
Sangoro, can I try? Okay.
Yes.
Because you're taking a group A separately and group B separately, so you later compare them.
Okay, thank you. Who else?
I can add that.
Yes.
So we are conducting a randomized control trial by putting a group of the group who receive intervention and the group that does not receive intervention to see the impact of the intervention. Yeah.
Yes.
Then I also say something about that.
Yes, Kokoda?
Can you hear me now? Because I have the internet issue. Can you hear?
Yes, I can hear you.
So I say randomization is important to reduce the bias.
Thank you very much. Yes. So randomization, let's say the importance of randomization is to reduce, Kokoda said bias, but it's to reduce the difference between the two groups. So if for example, if I came to Mikocheni in Dar es Salaam, I am, because the people who are in the same, I would say, proximity. If in the same proximity, then they probably have the same characteristics, yeah? Socioeconomic status, age group, they more often than not have the same characteristics. So the reason you do randomization is that you make sure that the people you pick, the people you pick are a bit similar, because you're picking them from the same group and they stay in a similar location, they're bound to have similar characteristics if you pick them randomly, yeah, as opposed to if you pick them according to socioeconomic status and stuff like that. So, but if you pick them randomly, then you reduce the bias or the difference between the two groups. And we will see why this is important. So you randomly pick two groups, what we call the control group and the intervention group. And it's called the control group because this group you do not give the intervention that you're interested in evaluating. The other group you give the intervention, and it's called the treatment or control group. And then you follow these two groups for a period of time, and then you compare the outcomes, yeah? So by virtue that you've picked them randomly, they're basically the same. And so the theory of this is that because they are picked randomly, they're necessarily the same, and the difference that you'll see in the outcomes would more likely be associated with the intervention. Yeah? Okay. If there's no question, then we'll continue. So that is why it is important to pick the groups randomly so that they're similar. And when you follow them with the intervention, without the intervention, at the end of the day, the difference that you see, you are more likely to associate it with your intervention. Okay, sources of data type. So we've seen methods of collecting data, but then generally they're usually sources of data, yeah? So where do you get this data from? So you want to go do cohort study. Where do you get the data? So this is what we call the sources of data. So there are two types of sources of data. We have the routine data, and this is data that is collected from health facilities. Routine because every time that a person goes to the health facility, the data the data is collected. So you can use this data because it's collected routinely. But then the data sources that are the data that are collected periodically. Let's say census. A census is collected, let's say every 2 years, every 5 years. A malaria indicator survey. We have something called malaria indicator survey. These are basically population-based surveys or data collection methods. So you go to the population and then you collect your data from the population. These are done periodically because one, of course, they are very expensive and they require a lot of logistics. Yeah. So these are the types of data sources. But that means that whatever your data source, you can conduct you can either conduct a randomized control trial, cohort study, observation from these data sources. I hope that is clear. And you've seen the difference between sources of data and methods of data collection. Yeah, so basically there are various sources of health data. We have health information system. This is data that is collected from the health facility surveys from health facilities, health demographic surveys, and then population-based surveys. So Juniper is going to put links to the different data sources that I've just put in my slide. Then you can refer to them later. So we have the DHS data. This DHS data is data that is collected from health facilities, and then it's aggregated at different levels. So at the ward level, council level, and a regional level. Yeah. So and then it's aggregated into something that we call a demographic, and it's aggregated by a software called Demographic Health Information Software. Yeah, which health facilities mainly use. And then population-based surveys are aggregated through something that we call DHS, Demographic Health Survey data sets. Okay, so you've collected your data. What do you do with your data? Yeah. After you've collected your data, you need to, because you need to tell the distribution, you need to quantify. Yeah. You cannot just collect data and then say, okay, now I've collected the data. Now what? So after you've collected the data, then you need to quantify the distribution of your disease. Yeah. So I went to Nkopeka. Sorry, it's stuck in my head. I went to Nkopeka. I collected. I did 200 interviews, and this is the number of cases that I have. So you need to quantify your data so that we know the distribution of the disease. So for example, various methods of quantifying your data are counts. Yeah, this is the simplest method. So I went and interviewed people in Nkopeka and found that they have leishmaniasis. Yeah, I found 50 people that have leishmaniasis. What does Okay, so I'll try and go a bit faster. So counts. So counts gives you the absolute number of disease that a person that you find in a community. The problem with counts is that it doesn't give you the baseline, the population. So for example, if you said I have 60 cases of cholera in Nairobi, what does that do? What does that tell you? Is it an outbreak? Is it what is the burden of disease? So counts never show you the burden of disease. You can say 60 cases of cholera out of what? Out of 8 million people. Then in that way you can see it's not actually an emergency. Yeah. But if it was 60 cases out of 100 people, then you could say there is an outbreak of cholera. So that is the importance of having a denominator in your calculation of distribution or in your quantification of disease. So we have counts, and then we have proportions. So number of people who have disease out of the total number of population that you sampled. So like I said, we have 60 cases out of 200. Then you can say the proportion of cholera in Nairobi would be what? 30%. Yeah. And then we have rates. Rates measures how often a disease occurs in a population over a specific period of time. So this one gives you how the frequency of occurrence of disease. So it shows you if the disease occurs quite frequently, then there is a problem, as opposed to if it occurs most slowly or occurs as expected. So for the measures of disease, we have counts, proportions, and rates. Anyone, can you think of examples of common measures of disease that you've come across? Neam, I'm sure you've come across some measures of disease.
Yeah, you put me on a spot. I'm trying to think. So I don't have a background in epidemiology, but when diseases or outbreaks have been reported, I think there are some metrics that they use. There's one around, I can't remember the technical terminology, but the rate at which the infected people die, or like the severity of it. I can't remember the technical term for it. But
All right,
that's
cool.
one that
Thank
I
you.
can think of.
Okay. Anyone else? Rates, measures of disease frequency? Yes?
Prevalence.
Thank you. Prevalence? Thank you. Anyone else? Okay, so for the purposes of this,
Mortality rates. Okay.
yes, okay, cool. Thank you. Anyone else? So let's see. Okay, so for the purposes of this workshop, we'll deal with two matrices, and the other matrices of disease, of course, like you said, mortality rate, death rate, and the other rates, yeah. But for the purposes of this workshop, we'll deal with the two major ones, which is prevalence. Prevalence is the number of people with disease out of the number of people sampled, yeah. So for example, here, and I'm going to ask, is Jacqueline around?
She isn't, sadly.
She isn't. So who's going? Can someone tell me what the prevalence of disease in this illustration is? So the disease are the people who have a bit of black. What would the prevalence be?
So
Harriet.
out
Yeah?
of 18, there's seven out of 18.
There is?
7 out of 18.
7 out of 18. So the prevalence. Cool. Thank you. Yeah. So prevalence is the number of people who have a disease over the total people sampled. And there are two types of prevalence. There's point prevalence that we said at a certain time point, at this exact time point, what is the number of people who have the disease over. Hello, everyone. So we're going to take up a talk from the measures of disease frequency. So there are two major measures of disease frequency that we use in epidemiology. Of course, there are others, but for the purposes of this workshop and the time that we have, we're going to talk about two measures of disease frequency. So these measures will be, the first one is prevalence. So prevalence refers to all cases of a disease existing in a given population, in a given population at a given time point. So we have two measures of prevalence. We have point prevalence, which is the number of all cases of a disease at one point in time in a defined population. So here what we need to understand is the definition of a defined population. So a defined population is a sample of population that you have set metrics for. So for example, a defined population is if you wanted to go assess or you wanted to go measure the prevalence of cervical cancer of women aged 45, of women aged 16 to 45 years in Kigoma district, yeah, for 4 weeks from the month in the month of August 2024. So that is your defined population. You see, you have set this population that it is only women who are aged 16 to 45 years of age. Where? In Kigoma district. And this is your population that you're going to measure prevalence from. So what you need to understand about prevalence is you need to have a defined sample population. Otherwise, you will not be able to describe your quantity. You won't be able to quantify your terms. If you just say, I'm going to measure the prevalence of cervical cancer in Kigoma district, what does that mean? Who are you measuring? Is it men? Is it women? Is it children? Yeah, so you have to have a defined population for you to be able to make inference from make prevalence quantifiable. Yeah, so we have two types of prevalences two types of prevalences. So we have point prevalence. So this is measuring the prevalence of a disease at a given time point. So the point can be you can take the samples from the women in Kigoma. It might take you 4 weeks, but it is at that time point that you have sampled all the women. It's the time it takes to sample all the women that you have defined in your population or in your sample population. Then we have cumulative prevalence. So this is a proportion of a population that has a disease at any time during a specified period of time. So for example, you go to Kigoma and you say, I want to measure the prevalence of cervical cancer between women aged 16 to 45 years of age from the month of August 2024 to the month of April 2025. So all the women that will have cervical cancer in that period of time are going to be the proportion of cases that you're going to measure. So it's all the people that have the case over a specified period of time. Point prevalence, it is at that particular point in time. I hope that is clear. And then we're going to go to our next measure of disease frequency, which is incidence. So incidence is the number of people with the disease who are a new case, yeah, over the number of people at risk that you have sampled. So our denominator is, of course, again, a defined population. So women age 16-45 in Kigoma district in the month of August, yeah. So this is our people at risk, yeah. And then you're going to find out how many of these people at risk, of these women aged 16-45, are a new case of cervical cancer. So what you need to remember here in incidence is that incidence uses person time at risk. And I'm going to try and explain what person time at risk is. So for example, let's say you are in, for theoretical purposes, let's say you go to Kigoma, yeah? And then your sample is 5 women aged 16 to 45 years of age who you're going to test for cervical cancer. So that is your sample population, five women. So let's say, for example, during your sampling, you find that 2 of those women, let's say your period was 1 year. So you're going to measure cervical cancer in Kigoma district for a period of 1 year from August 2024 to August 2025. So let's say hypothetically the number of women in Kigoma district were 5. So at the beginning of your measurement in August 2024, none of them had cancer. Then you continue measuring for 1 year. But then 6 months down so you test them maybe every month. 6 months down the line, you find that 2 out of the 5 women have developed cervical cancer. The 3 go throughout the year without developing cervical cancer. So if you want to measure person time at risk of all these women, you will say one woman was at risk for 6 months. and the other woman was at risk for another 6 months. So essentially, they were at risk for 0.5 years, and the other one was at 0.5 years. So the time that both of these women were at risk was what? It will be 0.5 months and 0.5 months. That is the time that these women were at risk. After they develop cancer, they're no longer at risk. Yeah. So how you're going to measure the prevalence of cervical cancer is you're going to take the time that each person in the sample population was at risk. So for the 3 women, how long were they at risk? 1 year plus 1 year plus 1 year, 3 years. And for the 2 women who developed cervical cancer, how long were they at risk? They were at risk for 0.5 years and 0.5 years. So your incidence will be measured in terms of 1 year, 3 years plus 0.5 plus 0.5. This is 4 years. So your person time at risk for all these 5 women would be 4 years. So your incidence rate will be measured by 2 cases, the number of people who had disease over the total time period of when they were at risk, which would be 4 years. So it would be 2/4, and your prevalence would therefore be the prevalence of cervical cancer in Kigoma district would therefore be 1 case. It would be 0.5 cases per person year. Yeah, because you'll get 2/4, you get 0.5. So, and it's measured in per person years. Your denominator is per person years, the period that each person was at risk. So the total period was 4 years. So it would be 0.5 cases per person year. Thank you. I hope that is clear. Next, we're going to go to measures of disease association. So measures of disease association quantifies the relationship between the exposure and the outcome, the disease. So the relationship between exposure and the disease occurring, and it also compares the measures of disease occurrence among the exposed and unexposed. So measures of disease occurrence is the prevalence and the incidence. In our case here, we've talked about prevalence and incidence. These are the measures of disease occurrence or disease frequency. So measures of association quantifies the relationship between exposure and outcome, and it also compares the measures of disease occurrence, this is prevalence and incidence, among the exposed and unexposed group. And there are 3 major measures of association. Of course, there are others, but for the purposes of this workshop, we're going to carry 3 major ones that you're going to come across frequently. So the first one is the risk difference. So the risk difference is the absolute difference in the risk of an outcome in one group, let's say the exposed group, minus the risk of difference in the other group. So the risk of difference here is it's basically probably, so let's say the risk of cervical cancer in women who smoke is 3/5. Yeah. So if you smoke, you are at, so 3 women who smoke have cervical cancer. So 3 women out of 5 who smoke have cervical cancer. So the risk would be 3/5. And the risk in the unexposed group would be 2/5. So that is what we mean by risk. It's basically the probability of you having a disease, having an outcome or a disease if you're exposed. And the risk difference is the absolute difference between these 2 risks. So it will be basically 3/5 minus 2/5. That is the risk difference. It just shows the absolute difference in risk between the exposed and unexposed groups. And it indicates the extent to which the disease can be attributed to the exposure. The relative risk. The relative risk is the ratio of risk of disease in persons exposed compared to the risk of disease in the persons that have been exposed. So back to our example, 3 smokers out of 5 people who do not smoke, 3 women who smoke out of a total of 5, group of 5 women develop cancer. So their risk is 3/5. And 2 women who do not smoke, do not get cervical cancer. So the risk of getting cervical cancer is 3/5. Yeah. That is in the exposed group. And in the unexposed group is 2/5. So the risk ratio, the relative risk is the ratio of risk in the exposed group minus the risk of the outcome in the unexposed group. So we are going to see this using examples. And then the odds ratio is the odds of disease in the exposed compared to the odds of disease in the unexposed. So an odds is basically the likelihood of an event occurring compared to the likelihood of the event not occurring. So in our example, the likelihood of an event occurring is 3/5. The likelihood of the event not occurring is 2/5. Yeah. So the odds would be 3/2. That is the odds ratio. Let's use examples here for me to try and elucidate it properly. So for example, let's say, let's start with risk difference and relative risk. We have a group of smokers and non-smokers, and we're trying to measure how the risk of smoking affects the development of coronary heart disease, or not, or affects whether you develop coronary heart disease or not. So let's say, for example, we have a group of 3,000 people who are smokers. 3,000 people who are smokers. So these are exposed. And out of these 3,000 people, 84 of them develop coronary heart disease. 219 of them do not develop coronary heart disease. So what is the risk of developing coronary heart disease if you're a smoker? So the risk of developing coronary heart disease if you're a smoker would be 84/3,000. Yeah, that is the risk. So you're basically at a risk. The risk is 84/3,000, and you get 0.028, and which is basically 28%. You are 2.8% at risk of getting coronary heart disease if you're a smoker. Yeah, if you're a smoker. If you're a non-smoker, yeah, what is the risk of you? So what is the risk of you getting coronary heart disease? So let's say for non-smokers, yeah, we have people who are 5,000 people who are non-smokers. 87 of them develop coronary heart disease, and 4,913 did not develop coronary heart disease. So what is the risk of developing coronary heart disease if you're a non-smoker? It is 87 divided by 5,000. So the risk of developing coronary heart disease if you're a non-smoker is 1.74. If you're a smoker, the risk of developing coronary heart disease is 84/3,000, which is 2.8%. So the risk of developing coronary heart disease if you're a smoker is what? 2.8. And the risk if you're a non-smoker is 1.74. So the risk difference is 2.8 minus 1.74. So the risk difference is 1.06. Yeah. So if you're a smoker, you are at a risk of you have an increased risk of developing coronary heart disease by 1.06%. Yeah. So you have an increased risk of developing coronary heart disease by 1.06%. Relative risk. So we found that the risk of developing coronary heart disease if you are a smoker is 2.8%. Yeah, and if you're a non-smoker is 1.74%. Yes. So the relative risk is the ratio between these two. The ratio of developing coronary heart disease if you're a smoker compared to the ratio of developing coronary heart disease if you're a non-smoker. And the risk, therefore, the relative risk of developing coronary heart disease if you're a smoker is increased by 1.60 is increased by 1.60 if you're a smoker compared to if you're not a non-smoker. Yeah, so you're at you are at a higher risk by 1.6. You are at a higher risk of getting coronary heart disease by 1.6 times if you're a non-smoker compared to if you're a non-smoker. I hope that makes sense. Then we go to the odds ratio. Yeah, what did we say the odds was? The odds is the likelihood of an event occurring compared to the likelihood of that event not occurring. So you're a non-smoker, you're a smoker. So what is the likelihood of you getting disease, coronary heart disease? Let's use 78 at this point. So the risk of you getting the odds of you getting the chances of you getting a disease, the people who get the disease, the coronary heart disease, if you're a smoker at 78 and not disease is 1,349. So what are the odds of you getting coronary heart disease if you're a smoker? We said it's the likelihood of an event occurring compared to the likelihood of the event not occurring. So the odds would be 78 divided by the chance the chances of the event not occurring. Yeah, the likelihood of the event not occurring. So what is the likelihood of the event occurring if you're a smoker? 78. If of which not occurring if you're a smoker is what? 1,349. So in the odds, we do not use the total population. We compare the likelihood of the event occurring to the likelihood of the event not occurring. Please remember that. And for the non-smokers, it will be the same thing. The likelihood of the event occurring if you're a non-smoker is 48, and of the event not occurring if you're a non-smoker is 6,129. So the odds of the disease occurring if you're a smoker is 78 over 1,349. The odds is 0.06. The odds of it occurring if you're a non-smoker is 48 over 6,129. So the odds is 0.008. So the odds ratio is the ratio of the disease occurring between the smokers and non-smokers. So between the exposed and non-exposed. And you see that the odds ratio is 7.5, meaning that if you're a smoker, you're at 7.5 higher odds of getting disease compared to if you're a non-smoker. Thank you. Then we're going to see how we interpret the odds ratio and the relative risk. So the thing that I forgot to mention is that relative risk is usually used when measuring incidence of disease, and odds ratio is usually measuring the burden or prevalence of disease. So how do you interpret this? So if the ratio is 1, so if the ratio between getting disease if you're exposed and unexposed is 1, that means there's no association between the exposure and the disease. So the chances of you getting the disease, whether you're exposed or not, are the same. So that means there's no association between exposure and disease. I hope that is clear. If the ratio is more than 1, that means that the people who are exposed are at a higher chance of getting the disease compared to the people who are not exposed. Yeah. So here we say that exposure indicates increased risk. So you're at increased risk of getting disease because the ratio, the measure of association, the ratio is greater than 1. If the ratio is less than 1, whether it's odds or risk ratio, if it's less than 1, that means that when the people who are exposed are at a lower risk of getting the disease compared to the people who are exposed. So it indicates a decreased risk if you are exposed to the disease. Okay. Then we're going to move to something we call hypothesis in scientific research. So a hypothesis is basically a statement or a theory about a cause of a disease or the expected outcome of an experiment or a test. So for example, and then a null hypothesis, let's talk about null hypothesis first. A null hypothesis is a statement that negates the hypothesis that you have come with. So it disapproves the hypothesis. So for example, let's use the case of John Snow. John Snow, his hypothesis was that contaminated water causes cholera. So the null hypothesis to John Snow's hypothesis would be contaminated water does not cause cholera. So there are 4 tests that we usually conduct to either approve or disapprove the hypothesis. So, and that is what John Snow was doing, to either approve his hypothesis that contaminated water causes cholera. So the first hypothesis, the first test, there are 4 possible, so the hypothesis test has 4 possible outcomes. You either fail to reject the null hypothesis when the null hypothesis is true. So for example, let's say the null hypothesis in John Snow's case was that contaminated water does not cause cholera. That is the null hypothesis, yeah? So if you fail, let's say in reality, actually, the contaminated water did not cause cholera. So if you tested and then you found that contaminated water does not cause cholera, so if you fail to reject the null decision that contaminated water does not cause cholera, this is a correct decision and we call it a true negative. So they tested and they found that contaminated water does not cause cholera. So the null hypothesis is true. So if you fail to reject it, which is the correct decision, then we call this a true negative. If let's say, for example, you fail to reject the null hypothesis, so the null hypothesis of contaminated water does not cause cholera. Let's say if you tested and somehow by some small percentage you found that your tests found that contaminated water causes cholera, but in reality contaminated water does not cause cholera. So the null hypothesis is true that contaminated water does not cause cholera. But if you reject the null hypothesis that contaminated water does not cholera and you go with the hypothesis that contaminated water causes cholera, this is something we call a type 1 error. So you reject the null hypothesis when it is true. The null hypothesis that contaminated water does not cause cholera, when that is true, but you go ahead and reject it. This is something we call a type 1 error, and you find a false positive of contaminated water causing cholera, while the reality is that contaminated water does not cause cholera. So that is a false positive. If you fail to reject the null hypothesis, when in fact the null hypothesis is wrong, so let's say the null hypothesis of contaminated water does not cause cholera is wrong, and the alternative hypothesis, which is contaminated water does cause cholera, is true, but you go ahead and fail to reject the null hypothesis. So if you fail to reject the null hypothesis, this means you accept it, that it's true, that contaminated water causes cholera, when in fact contaminated water does cause cholera, but you go ahead and reject this hypothesis. This is a incorrect decision, and it is known as a type 2 error. So you find a false negative. You find a false negative in the relationship between contaminated water and cholera. Yeah. So you say contaminated water does not cause cholera, when in fact contaminated water does cause cholera, but your finding was that it does not cause cholera. So you get a false negative. When you reject the null hypothesis, when the null hypothesis is wrong, so the null hypothesis, contaminated water does not cause cholera. So if you go ahead and reject this and say contaminated water does actually cause cholera, this is a correct decision, and we call this a true positive. And sometimes it is called the power of the test, which is the probability that you're actually going to reject the null hypothesis when the null hypothesis is wrong. Okay, let's look at this in the form of an example. So let's say a man goes to a hospital where he's told that he has cholera, but in that hospital they don't have confirmatory test, and he's told to go to another clinic for confirmation. So let's put this in hypothesis framework. Yeah, the hypothesis being tested is the true hypothesis is that the man has cholera. The null hypothesis is that the man does not have cholera. So we are going to try and fill up the table below correctly. So let's say the first reality is the null hypothesis that the man does not have cholera. Yeah? Yeah? So let's say the man does not have cholera. So, and then your decision, this is the reality. The reality is the man does not have cholera. And your decision your first decision is you reject the null hypothesis. So you reject the null hypothesis that the man does not have cholera when in reality he doesn't have cholera. So you have a false positive that he has cholera, while in fact he does not have cholera. That is what we call a false positive. If you go ahead and reject the null hypothesis. So let's say now the reality is he's found that he has cholera, and your null hypothesis is he does not have cholera. But if your decision is you reject the null hypothesis of he does not have cholera, when in fact he does not have cholera, that is the correct decision. Then our decision is, so the reality, go back in the reality here is he does not have cholera, and you fail to reject the null hypothesis, then that is a correct decision, because you fail to reject the null hypothesis when in fact the null hypothesis is true. So that is a correct decision. If you fail to reject the null hypothesis, which is that he does not have cholera, well, in fact, he does have cholera. This is a false negative. Yeah. So you say he doesn't have cholera when in fact he has cholera. So that is what we call a false negative. Okay. Thank you very much. We're going to stop there and take a 5-minute break for our next session. Thank you. So welcome back. So the second part of the workshop, as I had indicated earlier, is to do with malaria epidemiology. So we're going to learn basic malaria epidemiological concepts. Yeah, and in this section, it is expected that at the end of this session, you'll be able to define malaria and the global burden of malaria. You're going to be able to describe the general epidemiology of malaria, describe the history of malaria transmission and the controls that have been put in place. The history of malaria transmission control. You're going to be able to describe the global and local efforts that have been used to control malaria, and you're going to describe the different types of malaria interventions. So, malaria, what is malaria? Malaria is a parasitic infection that is transmitted to humans by the bite of a female Anopheles mosquito. It is caused by a parasite known as Plasmodium of the Plasmodium species. This is a parasite, and Plasmodium has 5 different known species. So we have Plasmodium falciparum, which is mainly transmitted in sub-Saharan Africa and causes about 99.7% of malaria cases in sub-Saharan Africa. Then we have Plasmodium vivax, which is predominantly transmitted in South America. So it causes about 74% of all malaria cases in America. We have Plasmodium malariae, which is found in Asia and South America. Then we have Ovale, which is also found in sub-Saharan Africa. So Plasmodium malariae, Plasmodium ovale, and Plasmodium knowlesi. These are not, they're not they're not transmitted in very high proportion. So they're parasites that are found very rarely. Yeah. So the prevalence is very low, if you will. Yeah. So most of the malaria cases that are transmitted globally are either transmitted by Plasmodium falciparum or Plasmodium vivax. So and Plasmodium is they're mostly transmitted by Anopheles mosquitoes. And there are about 3,500 species of mosquitoes, approximately 3,500 species. And of these 3,500 species, 30 to about 40 species are known to transmit malaria and disease. Yeah. So some of the other diseases that are transmitted by malaria would be what? Yellow fever, dengue, chikungunya, and Zika. We also have a species of mosquitoes that is called Culex, so not Anopheles culex, and these are known to transmit diseases like lymphatic filariasis and West Nile virus. Yeah, so in the context of our epidemiological triad, what do you think would be the infectious agent in transmission of malaria? So the infectious agent would be Plasmodium falciparum. And the host, it's either a human or a mosquito. And we'll see this ahead. We'll see the different types of hosts that malaria has. And this is what makes malaria very, very interesting. And of course, there is the suitable environment. So the host must be found in a suitable environment where he can interact with Plasmodium falciparum. And so since Plasmodium falciparum is an organism and it does not, there's no way that it interacts directly with a human being. It requires the Anopheles mosquito vector to transmit it from the source. So a source would be a host that is infected to another host that is susceptible. And in between here, it needs the vector Anopheles mosquito to be able to transmit the vector. So as I had said before, as we had mentioned before, that there are two types of transmission, the biological transmission and mechanical transmission. In the case of malaria, the transmission is biological. So the Anopheles mosquitoes, Anopheles mosquito is defined as the definitive host of the malaria parasite. Why the definitive host? This means that it is the host where the parasite or the microorganisms undergoes sexual reproduction, reaches sexual maturity and undergoes sexual reproduction. So, and we will see ahead how this occurs. And in malaria, the human is also we would say an intermediate host here. So the Plasmodium falciparum needs to undergo some development, not sexual. We call it a sexual development in the human in order to complete its life cycle. So the mosquito picks the parasite from an infected host. It undergoes a sexual reproduction in the mosquito, and then it is transmitted into the susceptible host, where it undergoes another development, another transformation before it is picked up by another infective bite of a mosquito. We will see all this ahead. We'll explain the asexual and sexual cycles of the malaria of the Plasmodium falciparum. So endemicity of malaria or burden of malaria. So malaria is basically endemic in sub-Saharan Africa and in Southern Asia and South America, Southeast Asia and Southern America. So for example, for the distribution of malaria in 2023, there are approximately 263 million cases globally. And sub-Saharan Africa had about 246 million of these cases, and there are about 569 global deaths. So, and sub-Saharan Africa experienced about 76% of all global deaths. And malaria also disproportionately affects children under 5 years of age. So in 2023, it was also about 77% of the deaths that occurred in under fives in Africa, was for under fives. Central and Southeast Asia account for about 4 million cases of malaria. That is about 1.5% of the global burden, and 48% of the global P. vivax malaria infections. And then Southern America contributed about 0.5 million cases of P. vivax. So that is the global burden or distribution of malaria. We can see that Sub-Saharan Africa is disproportionately affected by high malaria cases as compared to Southeast Asia and Southern America. Globally, malaria transmission has been declining. So from we like to say from 2000, but I personally think it's earlier. There's been a great decline in malaria, mainly because of the interventions that have been put in place and intervention and control measures that have been targeted towards malaria. Another thing is that in time, malaria would go down, even despite there being interventions. Malaria would go down because of development, natural human development. Yeah. So, for example, because of development such as in towns and cities, this would destroy malaria habitats that would be found predominantly in areas that are not developed. Yeah. So in this way, malaria would have naturally go down. But then, in addition to going down naturally, the interventions have pushed the malaria further down. So, for example, in 2000 to 2000, from 2000 to 2015, there's been a steady decline in malaria. Yeah. And then from 2017 to 2022, we see that there's been a bit of malaria stability. So there hasn't been very great declines in malaria as compared to from 2000 to 2015. And then from 2022 onwards, we can see that there have been reported cases of rashes in malaria. For example, as I said earlier, there are 263 million cases. This was an increase of about 11,000 cases compared to 2002. Yeah. And also in the Sub-Saharan Africa in 2023, there were 246 million cases up, and these were higher than the 237 million cases that were observed in 2022. The same thing, and the deaths from malaria has remained relatively stable. So there have been about 569,000 deaths in 2023. And this is only a decrease of about 4,000 in 2023 compared to 2022. So we can see that there hasn't been great decline, and this is because I would say the interventions that are used for malaria control are reaching their definitive biological limit. So the interventions that are being used currently cannot lower malaria any further. So we need to come up with innovative ways to impact the remaining foci, the remaining areas where malaria is being transmitted. And this is, of course, as a result, like we said, those high malaria transmission, let's say from 2000. So malaria was endemic everywhere. And because of the high malaria control, extensive malaria interventions that were employed from 2000 to 2015, where malaria was endemic everywhere, this had a significant reduction in malaria cases or malaria burden. But then as time has gone by, then malaria has declined, such that in areas where malaria was previously endemic is now, malaria has declined to be spotty transmissions. Yeah. So it is what we call heterogeneous, where it is no longer a stable endemic transmission. We just have places where malaria, we have foci where malaria is transmitted because of the extensive intervention measures that have been put in place since 2000. And as a result, this malaria becomes very hard to control because you don't know where to target your interventions. You cannot say let's still use nets everywhere, because the areas where the nets would not work. Malaria, first of all, malaria has been reduced to such low levels that even though you use the nets, then you would not have any, you would not see any impact. So the ideal situation is to isolate these areas where malaria is heterogeneous and target them, specifically target those areas where there's still high malaria transmission, as compared to a blanket interventions of nets where you would put nets in areas where nets are not needed, and therefore you divert the resources that would have been, if you had targeted the malaria to the regions where there's high malaria transmission, you'd have more effectively used your resources. Yeah, so that is still on global malaria burden. So we see that, like I've said, WHO Afro countries or sub-Saharan African countries disproportionately suffer from malaria burden. And like I'd mentioned before, sub-Saharan Africa is home to about 94% of all global burden malaria. Yeah? And in sub-Saharan Africa, there are just about 11 countries, yeah, which are responsible for about 66% of the global malaria burden and 68% of the global malaria deaths. And these countries are called high burden, high impact countries, yeah. And they include countries like Burkina Faso, Cameroon, DR Congo, Ghana. We have India, Mali, Mozambique, Niger, Uganda, and Tanzania. So these countries contribute to about 66% of the global malaria burden. And therefore, it is prudent that if we want to have an impact of malaria, then we focus on these countries that have high burden of malaria. And that is how the WHO came up with a strategy called high burden, high impact. So you target these high burden countries, and then you're bound to see a high impact of your interventions. So as I mentioned earlier, there has been historical malaria control measures, malaria control measures that have been implemented historically from the 1950s to up to today, to 2024. So, for example, this figure just shows the different malaria control strategies that have been put in place. So, for example, so initially there wasn't any coordinated malaria control efforts until after the WHO was formed in 1948. This is where we had coordinated malaria control efforts globally. So from 1950s to the 2004 is where we have seen coordinated malaria control efforts. And this starting with 1951, where the United States Malaria Eradication Program successfully eliminated malaria as a significant public health in the Americas. Yeah. And then in 1955, the WHO launched the Global Malaria Eradication Plan, whose aim was to eliminate malaria worldwide through extensive use of DDT. DDT is a chemical that kills mosquitoes, and chloroquine. Chloroquine is a drug that was used to treat people infected with malaria. And then from 1961, so these are just significant events that have taken place in malaria control. So from 1961, people started realizing that the malaria parasite, Plasmodium falciparum, was actually becoming chloroquine resistant, and they needed to come up with other drugs to treat malaria. So in 1961, the first reported cases of Plasmodium falciparum resistance to malaria was reported. Yeah. So, and then in 1969, after about 7-8 years of WHO efforts to control malaria, the WHO abandoned the Global Malaria Eradication Plan. This was mainly due to logistical challenges, which meant it was expensive, there was increasing resistance to insecticides such as DDT and drugs such as chloroquine, and therefore it was difficult to sustain these programs. They couldn't put out the control efforts that they were using, yet they knew that there was resistance to DDT, the insecticides, and the chloroquine drug that they were using. So they abandoned this malaria eradication plan for about 20 years, which so up to about 1992 is when the WHO also came back again with global malaria control strategy, which shifted all focus from eradication, which was an ambitious plan from the global malaria eradication plan, which was an ambitious plan. So they shifted the focus from eradication to control through improved diagnosis and treatment. So improved diagnosis is come up with better methods of diagnosing malaria. And it's around this time that RDT was developed, and therefore, and other drugs like the triple therapies. First of all, it was of course the sulfur pyrimethamine, or SP if you want. And then in 1998, the RBM was formed, so Rollback Malaria Partnership was formed, and it was established by WHO in partnerships with other global organizations such as UNICEF, UNDP, and World Bank. And the major role of the Rollback Malaria Partnership was to have the malaria deaths by 2000 through global coordinated malaria efforts. So, and it was around this time where after rigorous controlled, randomized controlled trial, as I mentioned before, where ITNs were endorsed as a tool for controlling malaria. Yeah, it was around 1998 to 2000. And therefore, as a result, there was increased funding to distribute infectious treated nets to globally, especially to sub-Saharan Africa, which suffered more malaria burden compared to other areas. Another significant event in malaria control was in 2000, where we had the Abuja Declaration, which was signed by about 50 African leaders who committed to reducing malaria mortality by about 50%, and they were going to do this by each country contributing funds for the control of malaria. So the aim was for the Abuja Declaration was to reduce malaria mortality by 50% in 2010, and in addition to the funds that were given by the RBM partnership to control malaria, the Abuja Declaration African leaders also gave some funding for control of malaria, and this saw an increase in funding and political commitment for malaria control that had been unprecedented before. And in 2005, we had the President's Malaria Initiative program launched, which boosted the US support for malaria control in Africa. So this included funding things like ITNs, these are bed nets, and indoor residual spraying, and the ACTs, which are the triple therapies that are used for treating malaria. Then in 2008, we saw the Global Malaria Action Plan developed, and it was launched by the RBM partnership, whose goal was to reduce global malaria cases by 75% and eliminate malaria from 8 to 10 countries by 2015. And in that 2015, because the RBM partnership did not realize its goals of reducing malaria by 75% and eliminating malaria in 8 to 10 countries, the WHO adopted another strategy, which is called the Global Technical Strategy for Malaria, from 2016 to 2030, whose aim was to reduce malaria incidence and mortality by at least 90% in 2030. So we're still under the Global Technical Strategy for Malaria Control. although we are still following this strategy, I think as is being reported, the goals for the GTS by 2030 will look unlikely to be achieved unless we have more funding. As you've realized, some of the funding for malaria control was diverted in 2019 to combat COVID. So unless there's sustained investment and innovation and expanding, there's expansion of access to prevention and treatment, it is very the malaria community is very skeptical that the GTS goals of 2000 and of 90% reduction by 2013 of malaria cases will be achieved. So that is just a run through of how malaria control has been from the 1950s. So to talk about the malaria life cycle. So the malaria parasite is a very interesting parasite. It undergoes both sexual and asexual reproduction. So for the sexual phase, this is called the exogenous phase, and it undergoes reproduction in what in the mosquito, what I had earlier alluded to as the definitive host, and this process is called sporogony, the sexual reproduction that goes on in the mosquito, and then it also undergoes an asexual phase in the human host, who is the intermediate host, and this process is called schizogony. So I'm going to try and I'm going to explain to you the first the asexual reproduction, and then we're going to go to the sexual reproduction of the malaria parasite in the mosquito. So what occurs first is that an infected mosquito, so a mosquito infected with malaria parasites, bites a human host, and then it injects what we call the sporozoite form of the malaria parasite into the bloodstream of the human host. And then the sporozoites migrate through the bloodstream to the liver of the human, to the liver cells. These are called hepatocytes. And then in the hepatocytes, the sporozoites undergo something called schizogony in the liver cells to produce, so they split up to produce, they undergo transformation to produce something called a phase called the merozoites. So the merozoites then develop in the hepatocytes, and then after a while, they burst the hepatocytes, these are the human cells, and they're released into the bloodstream. Yeah. And when they're released into the bloodstream, they then infect the red blood cells. The merozoites infect the red blood cells. So when they infect the red blood cells, the merozoites develop into something called a trophozoite, which is a ring stage. You'll hear a lot something in malaria world, people referring to the ring stage. Yeah, so it develops into a ring stage, and then it multiplies into it multiplies while within the red blood cells. Yeah, so it divides asexually in the red blood cells, producing more merozoites, and therefore when after a while, the merozoites in the red blood cells then burst the red blood cells and are released into the blood. So when the merozoites are released into the blood cells, they either infect new blood cells, or they can either develop into male and female gametocytes. Yeah. So the merozoites will either infect new red blood cells, or they will develop into a portion of them will develop into male and female gametocytes. So when they develop into the male and female gametocytes, then the mosquito, another mosquito, then bites the infected human and ingests the gametocytes. Yeah. So the male and the female gametocytes then fuse while inside the mosquito to form something called a zygote. Yeah. And then this process occurs in the digestive tract of the mosquito. Yeah. They fuse to form a zygote, and then after a while, the zygote then the zygote then produces what are called sporozoites, yeah? Then the sporozoites burst from the midgut of the from the digestive tract of the mosquito, and then they travel to the salivary glands of the mosquito, where in the salivary glands the sporozoites are injected to another susceptible human host. So this, in short, is how the mosquito, the Plasmodium falciparum life cycle goes through the humans and into the mosquito, and from the mosquito into the humans. So one major thing to note is that when the merozoites burst from the red blood cells after the liver stage, when in the blood stage, when they burst from the red blood cells, this is what causes the fever and chills. When you hear someone has fever and chills, that means that the merozoites have bursted from the red blood cells and now are in the bloodstream before they infect another red blood cell. So this is what causes the fever and chills when someone has malaria. So that is just something to note. If there are any questions, since this is a recording, please feel free to get in touch with me and I will explain more. Then after that, then we have a few exercises. So in these slides, for this part of the recording, will not be shared with you, but we would like you to list agent factors, so Plasmodium factors that you think are to list. And then also we'd like you to list the host factors that you think affect, the human host factors that affect malaria transmission. And lastly, for your exercise, we would like you to list the environmental factors that affect malaria transmission. So we will go through this our next, we'll go through this in our next workshop before we get into the grind of the next workshop. Lastly is the diagnosis of malaria. So what do we use to diagnose malaria? So there are several ways which malaria can be diagnosed. So we can diagnose malaria through microscopic means. So this consists of examining the blood specimen of someone who's suspected to have malaria under a microscope for malaria parasites. So the malaria parasites have a certain morphology that is different from the red blood cells. So if you look at them under the microscope, then those entomological lab technicians who know, they can tell if there are parasites actually present in the blood specimen and how many there are. So microscopic means is one of them. Then there are serological tests for malaria, where antibodies and antigens produced by the body's immune system against the parasite can be detected in the blood. And of course, the RDTs, which are based on detection of circulating parasite antigens in the blood. Quantifying malaria. So we've seen the ways of quantifying malaria. We looked at prevalence and incidence. But for malaria, there are two major matrices that are used for quantifying malaria. We have the annual parasite incidence, which is the number of new confirmed malaria cases occurring in an area during a given year or during a period given of time, a given period of time, over the total number of people that were under surveillance. So over the total number of people that were sampled, that were tested for malaria, how many new cases of malaria, how many new cases of malaria did you find? And then we have the Plasmodium falciparum parasite rate, which is just the confirmed cases of Plasmodium falciparum in 1 year over the total population in surveillance. Yeah. So Plasmodium falciparum parasite rate is a form of malaria prevalence, parasite prevalence, and annual parasite incidence is a form of malaria incidence. The global technical strategy. So this is just these are just the strategies that are recommended by the Global Technical Strategy, which, as I had mentioned earlier, is the strategy that the WHO and the malaria community has adopted from 2016 to 2030. So with the aim of reducing malaria cases by 90% globally in 2030, they've endorsed the following, I would say, pillars or strategies to be able to reach the goal. So one is to ensure universal access to malaria prevention, diagnosis and treatment. So malaria prevention would be universal access to drugs, access to diagnosis, let's say ADTs, and treatment when you get malaria. And then, of course, accelerate efforts towards elimination, attainment. So in areas where malaria transmission has been reduced to low levels, you can target focal areas where there's still high malaria transmission to drive them even lower to be able to eliminate malaria in such areas. And of course, transform malaria surveillance into one of the co-interventions. So initially, malaria surveillance wasn't a co-intervention of malaria. It was just surveil and say malaria is here. But what the GTS is proposing is that we continually surveil for cases of malaria in areas where there's low prevalence, and in high prevalence areas we can control malaria until we reach low transmission areas where we can start thinking of elimination. Another one is, of course, harnessing innovation and expanding research. So harnessing innovations, like I said, most of the contemporary malaria control tools have reached their biological limits. So we need to come up with innovative ways in which we can further drive down the existing foci of malaria. And of course, we can also expand malaria research. So malaria research will help us come up with innovative ways for controlling malaria. Then we have the primarily malaria control and prevention measures. So malaria control primarily focuses on reducing the overall reservoir of or reducing the overall population of people that are infected of malaria, and therefore reducing and in that way reducing the proportion of mosquitoes that can carry the malaria parasites. And it also focuses on reducing the contact between the host and the infectious agent. So you can do this through things like chemoprevention. So chemoprevention is just like the word says, is prevention using chemical means. So it suppresses and prevents infections in humans through things like giving malaria drugs. So at the combination therapies, so artemisinin combination therapies. You can give intermittent preventive treatment to women, so IPTI. So you give drugs like sulfur pyrimethamine to women to prevent them from getting malaria. And we also have things like seasonal malaria chemoprevention, where you go to an area of high malaria prevalence, and in a season where let's say after the rainy season, where you expect to be a spike in malaria transmission, and you give all these people drugs that will prevent them from contracting malaria. Then we have things like case management, where if a case comes to a hospital, of course after diagnosing and treating, then you can diagnose and treat, and then follow up the person in the community from where he came, and then you can test around to see if the other community members around that focal case that are present in the community, and then you can treat this as well. So this is just another way of controlling malaria here. We have prevention of malaria, which primarily focuses on reducing human vector contact, and we do this through things like insecticide-treated nets, indoor residual spraying. So insecticide-treated net is where you use a net so that an infected mosquito cannot, if you sleep under net, an infected mosquito cannot come into contact with you and inject Plasmodium falciparum parasites inside. We have indoor residual spraying, which also prevents contact between the mosquitoes and the human beings. So indoor residual spraying is where you spray the walls of human dwellings with an insecticide, so that when the mosquito comes into contact with that insecticide, then it dies. So it reduces the population of mosquitoes that have a Plasmodium parasite. And in this way, it also reduces the number of mosquitoes that can infect you, in that way, driving malaria down. And then we have malaria surveillance, which is to detect and investigate all malaria cases. And like I said, look for secondary cases from the primary case. So a person comes to the hospital, treated, and then he's followed up to his community, and therefore he's tracked back to his community, and all the people that are within the vicinity of that case are tested for malaria, and if they're found with malaria, they're treated. So those are control measures, and then there are complementary tools that can be used for malaria prevention. I repeat again, as I mentioned, that the current control efforts, ITNs and IRS, indoor residual spraying, are reaching their definitive biological limit. So they've reached a limit where even if you increase the access to ITNs and IRS to 100%, you will not be able to reduce malaria any further. That is what I mean by saying it has reached the biological limit. So in such a case, then it calls for introducing or coming up with other ways in which can impact the malaria transmission in areas where the contemporary methods or the current methods of malaria control cannot have an impact. So, for example, there are some malaria mosquitoes that bite outdoors. If you distribute ITNs to 100% of the people in that community, they will only use the nets when they're indoors. When they're outdoors, they're not protected. Yeah. So you see, in such a way, the ITNs have reached the biological limit of protecting the humans, in that they only protect the human host when it's indoors. So we need to come up with other innovative ways which can attack the mosquito when it's biting people outdoors. So some of the methods that have been developed are things called odor-baited traps or spatial repellents. So odor-baited traps are traps that are fused or are fused with chemicals that smell like humans. So because mosquitoes are attracted to chemicals produced by humans, they go towards this trap, and when they land on this trap, the traps are treated with insecticides. So when they land on these traps, then they pick up the insecticides and die. We have spatial repellents. So I think all of you know about topical repellents, but now we have spatial repellents. So a spatial repellent is a repellent that produces, if you for lack of a better term, a bubble around the person that is using it. So when a mosquito comes towards a human who has a spatial repellent, then it is repelled, yeah, before it reaches the human host. Of course, we have things like biolarvicides, which are put into malaria mosquito breeding habitats. Therefore, they prevent the mosquitoes from reaching the mature stages. We have things like zooprophylaxis. So the mosquitoes that I would say opportunistic, yeah. So if the human host is not available, let's say is under net, then they can survive by biting domesticated animals. And these mosquitoes have also been found to be able to transmit mosquitoes. So if you can target animals that these mosquitoes bite, then you can reduce the population. And in this way, also reduce the population of mosquitoes that can potentially transmit malaria. We have things like spraying swarms. So when mosquitoes are mating to produce eggs, which is the major reason why mosquitoes bite humans for egg development. So when mosquitoes are mating, they usually form a swarm. So the malaria world has come up with sprays where you can spray the swarms where they're mating. So in this way, you prevent matings. In fact, the mosquitoes cannot if they're mated, then they will not need the blood for the eggs to develop in the mosquitoes. We have attractive toxic sugar baits, so mosquitoes usually depend on sugar from plants for energy. So if you bait this sugar with toxic chemicals or insecticides, then when the mosquitoes goes to feed on the plants, then they pick up the insecticide and they die. So all this is just to reduce the population of mosquitoes that can potentially transmit malaria. The challenges to malaria control. So we have things like climate change and destruction of the natural environment. So if you destroy the natural environment, then you expose the areas where you expose where mosquitoes could not reach human beings, but by destroying the natural environment, then you expose the human beings to the mosquitoes. So poverty and substandard health services, I think we've talked about this. If you cannot access health services to be treated, then you can transmit malaria and potentially it can be fatal for you. Cross-border malaria transmission. So there are countries where malaria has been controlled very well. So for example, Eswatini in Southern Africa, they have a very good malaria control system where they experience, I would say, about 6 malaria cases per year. But neighboring them, the neighboring countries, in Southern Africa, Zambia, Zimbabwe, Mozambique, they do not have very robust malaria control systems. So if a person moves from Mozambique, comes into Eswatini, where malaria has been reduced to such low levels, such that the immunity for malaria has gone down in the population, then they can easily infect the population and can easily cause an epidemic. Of course, we've talked about resistance to antimalarial drugs and insecticides, and the other one was decline in funding. So, for example, we saw during COVID, the funding that was meant for malaria control was diverted to the donor countries to be able to handle their COVID cases. And just recently, as last week, we've seen that President Trump has cut funding to donor funding to countries that depend on funding for control of diseases. And this is likely to have a great impact on malaria control. So I think that is all I have for malaria epidemiology. If you have questions, then you can ask Juniper or you can ask, I am available. We are all available. So if you have any questions, please reach out to us. Thank you very much.